Okay, from the last class, we're looking at the stages in micropropagation. What how many stages? What are all the steps we need to take? The steps we need to take for micropropagation. All right. So this class will have example of micropropagation in banana and also in an ornamental crop. That would be rose. Right. So we have. So how many stages we have? ID number 1717. How many stages are there in micropropagation? 0 to 4. Okay, that is enough. So you have 5 stages, right? 5 stages. So this will be in 5 mark question always. Stages in micropropagation of mostly the banana micropropagation. So you have 5 stages. So starting from 0 to 4. So this is an overview of any plant tissue culture lab, how neatly that needs to be maintained. So the different parts of the lab, starting from the first, okay, so there's no washing room here. Right, so for the first one is the washing room we need to have, then the media preparation room, then the transfer or inoculation room, then the culture rooms, the growth room, the culture racks, then for hardening, you have the weaning chamber and the growing area, and before field transplanting or marketing, that goes into the holding area into this open field. So this would be the normal procedure for any of those crops for micropropagation. So the first banana, the most say marketed crop, tissue culture marketed crop is banana. So the India leads the in the world banana production with about 30 million tons. Right. So the India is the largest producer, and the second is China. So between China and India, there is 20 million tons difference. So China produces around 10 or 12 million tons, but India produces 30 million tons, and the majority of it is being consumed internally. So it's not being exported. Very less amount is exported, and banana occupies 1/3 of all area among those fruit crops. Right. So much of this area is occupied by banana. So for that we need to. So before selling the tissue culture plants, we need to have the certification systems, right? NCSTCP system, so that it is virus indexed with no diseases. So that needs to be sold. Then the highest production is in Maharashtra, and the second followed by Tamil Nadu. And Maharashtra also has the highest productivity of 65 metric tons compared to the national average, or in Tamil Nadu also, we get only half of what we get in Maharashtra. So this is the importance of banana, and conventionally, normally swat suckers. So these are all those swat suckers. Swat sucker in the sense the leaves resemble like the sword, right? So they don't open up until the plants get much larger. So these are all those swat suckers. So this will be the water sucker. So water sucker is with broad leaves. Mostly this is used for leaf purposes. So இப்போ வாழை இலையெல்லாம் இதுல இருந்து எடுக்கக்கூடாது. So if you cut each and every leaf from banana, each and every hand is the bunch. What we have is called as the bunch, and each banana will be called as the finger, right? So in banana bunch, you have the hand, and each banana will be called as the finger. So if we cut one leaf when it is growing, if we cut one leaf, so each hand will be reduced, right? Over seep. So the என்ன சீப்பு சொல்லுவோம், சீப்பு அதில் ஒவ்வொரு வாழைப்பழம், right? So if we cut one leaf, one seep, right? One hand will be reduced. So we need to be having, we should not cut leaf from this, that is for the fruit production. So mostly for the leaf, they use this type of plants or suckers. Right? It's mostly swat suckers are being used, and suckers mostly can be infected with pathogens. So the most important pathogen is the fusarium wilt of banana. Right. So the next, this fusarium wilt is also coming up to this coconut now. Right. So most of this coconut is being infected with fusarium, and there is very less control measures we can take once it is infected. So, and if we use the conventional methods, so there will be no genetic uniformity. So there can be various stages. So when we look at the suckers, so this is of the much younger age, this is of much older age. So when we use different age suckers, so each and every time the maturity, so the bunch production will also be delayed. So we need to be have a delayed harvest. So harvesting in normal conventional sucker field. So the harvest can go up to 1 month difference. So that for that we cannot have laborers for the entire 1 month for the harvest. So if everything comes for harvest at a time, then we can have laborers so that we can harvest at one time, right? So that is one advantage of having tissue culture plants. So this here you will be having again the harvesting is prolonged due to the variation in the age and size of the sectors. So the advantage will be to use the tissue culture plants where there is higher rates of multiplication of elite clones or plants, then production of disease-free planting material. Record எழுதிட்டு இருக்கேன். இங்கிருந்து பார்த்தா தெரியாது. What record is it? Close it. So here we can have this production of disease-free planting material, then small amount, all the advantages of going in for tissue culture. Small amount of space is required for multiplication for a very large number of plantlets. And the plantlets what we get will be of uniform size and age, so that all the plants will come for harvest at that time, right? At the same time, and the harvest will also be much easier, and high quality of fruit bunches, and the plantlets can also be available throughout the year, and whenever we need it, we can immediately produce a large number of suckers for planting. So, the suckers in banana. So mostly, so this will be the SWAT sucker. And whatever we get, we need to select this type of SWAT suckers, not the water suckers, right? So this water sucker, they have large and broad leaves right from the emergence. Immediately after germination, it will have a broad leaf or expanded leaves, and also very small rhizomes. And not recommended. They produce inferior fruits. So what we need to select is this type of the SWOT suckers. So where the leaves emerge only after the stem, the pseudostem reaches a certain height, and it will have a broad, large rhizomes and used for rigidity propagation or like continuity of this banana planting, which gives rise to this quality planting material. So the stages, different stages. We need to take as we get this the rhizome. So each of those rhizomes. So these are all the worlds. You know, the worlds of the rhizome. Also, there are you have the sheets, and where inside this, in the middle of this, you have this meristematic or the shoot tip. So what we need is the inside of this. What we need to remove all that, right? So this banana. Why we need to remove this all the sheets to access this the meristem or the shoot tip. So inside it will be the shoot tip. We need to get the shoot tip of banana, right? So in the banana. So why the shoot tip is in the rhizome, not in the top of the plant? So what's the stem modification in banana? Huh? Pseudostem. You have pseudostem. It is a pseudo. Pseudo is false, right? False stem. So why is it false stem? So where's the actual stem? A false stem அப்படின்னா duplicate stem அப்படின்னா original stem எங்கேயாவது. So where is the original stem? Practicals, have you finished this in practicals? All right. Huh? Okay. So an actual the stem modification in banana is called fasciation, right? Fasciation is compression of the stem, compression of the entire stem, right? So when you have, say like when you have the entire stem, right? Entire stem, you have the nodes and evenly placed nodes, and you have the internodes, right? So if you compress this entire stem into a round ball, so what will happen is all these nodes will be also be compressed, right? So this the entire thing will be compressed into a very small structure. So for all those nodes and internodes will also be compressed, right? So when we remove all this, when we cut all this, and whatever we get, the pseudo stem is the extension of all those nodes. So nodes இருந்து எப்படி leaf வருதோ, that will be the petiole that forms the actual stem, right? So okay, so the next class when we get into this the practicals, yeah, you will see when you cut it, you will see those striation code, right? So those will be the nodes where you can have this the axillary buds, right? So the axillary buds. So normally in banana, so this entire, the entire because the shoot tip is inside, the shoot tip controls all other side branching, right? Because of apical dominance. So because that controls all other side branching, so there will be no side branching in banana, right? So when you cut it, when you remove all this, when you remove all the sheets and get into the inside of this pseudo stem, it's okay. உங்களுக்கு தெரியுமா? So this stem, and you cut all this, you will see the striations. Where you will have this, each of them axillary buds, right? Each of this axillary buds controlled by all of this, the actual shoot tip. So this shoot tip controls all this buds. So there can be no other side shoots coming up when this is being controlled by all this. But this will also produce the leaf, which will be the petiole, which goes up, and this will give the actual leaf. Right? So when you take out, so you remove all this, so you cut all of this, shave all of this, and get only this part. Right? So you get only this part. So when you take this part alone, to the shoot tip, and there are no other because this controls all other side branches, right? So how many shoots will you get from this? You put it together. So micropropagation is for producing large number of planters, right? So when you put one shoot, you get many number of shoots here. So how will you get many number of shoots? From the buds you need to get. So each of those buds needs to give a plant, but normally it doesn't give. Banana, right? Yes or no? Banana, you get only one plant out of this. You don't get any of this because of this apical dominance. So here, when you put it in the media, you get many number of shoots, like many shoots. How do you get many shoots? You should get only one. Which batch did this tissue culture? You get A batch there. ID number 6. 6, 7. So what did you do before putting onto the medium. Ascorbic acid. Why ascorbic acid? Okay, browning. Now, what we are talking about? Many bugs. Simply வரலாம். Many bugs ஏன் வர மாட்டேங்குது? Apical dominance. அப்ப என்ன பண்ணணும்? Apical dominance destroyed. So you gave a vertical cut. Did you? Yes. Why vertical cut? Multiple shoots. So the cut பண்ணி வச்சா வந்துருமா? Just a vertical cut, not splitting of that. Just a vertical cut. So you get you reach to this meristem and you destroy the meristem. So when you destroy the meristem, there will be no apical dominance. So you get many number of shoots, multiple shoots, all from the side axillary buds. What you see. Right? So you get multiple shoots. The stage 0, the selection, then sterilization and all those, then inoculation into the media. Then stage 2, multiplication of the stools. Stage 3, in vitro rooting. Then stage 4, the commercial production, the hardening. Right? So you have 5 stages starting from 0 to 4. So this, right, okay. So you have the banana fruiting bunch. So you get the rhizomes trimmed rhizomes, and whatever you see in the middle. So this will be the shoot apex, right? Growing shoot tip. This is what you need to take out, and you give a vertical cut here to the shoot tip wherever you are planting onto the medium. So this will destroy the meristem and also the apical dominance. So you can get. So you see, maybe I don't know if you can see those striations. Right? So all those strings. So this will be the compressed node, right? Compressed node. So you also have that in tulips or this colocasia, the கருணக்கிழங்கு இதெல்லாம் இருக்குல்ல கிழங்கு எல்லாமே. So those are all those fasciated stems. So where you get all those striations and also the nodes from which you can get those axillary buds. Right? So you get multiple shoots, rooting, and also hardening, and also transplanting into the field. So this, okay, the banana bunch when transplanted into the field. So this will be the entire bunch, and each individual will be called as the hand, and individual fruit will be called as the fingers of the banana. So the planting material would be the swat suckers weighing around 1 kg, or commonly used as the propagating material. So you cut it, trim it, and all those sheet around that needs to be removed. So you get only this this kind of swat suckers, right? Not broad-leaved suckers. You remove all other. So you give a vertical to the mother plant. So you get this the swat suckers. So the suckers you remove each and every layer of that. So for each and every layer, removal of the layer. So this can be done outside. So remove those layers and then start the surface sterilization. And then for each and every removal of those sheet, go in for sterilization, and finally you will reach a stage where you cannot remove any more of those sheet. So that can be used for planting into the medium. So the media, the factors affecting this plant micropropagation is the growth medium, whatever we are going to use, the environmental factors, the conditions, the explant source, and also the genetics of the plant. So what type of plant we are taking up. So the common media, as usual, MS media with the 5 mg BAP. And for shoot elongation, BAP 2 mg and IA 0.5 mgs, and rooting. And mostly for the rooting, we need to eliminate all the nutrients. So if we go on giving the nutrients, so the plants need not produce the roots, so will not produce roots. Right, it will be taking up the nutrients from the medium. So in order to stimulate the plant to produce roots, so reduce either quarter or half ms, and this auxins, IBA, NAA, and also some activated charcoal so that there is no browning and hardening of those plantlets that is produced. It's agar. The agar is washed and placed in the mist chamber with the cocoa peat and then in the shade house for a secondary hardening with sand, red soil, and all this. Now the hardening can go on for 15 to 45 days in the mist chamber for hardening. So an ideal tissue culture raised plant should be around at least before marketing. For marketing, it should be at least 30 centimeter in height and have and should have a pseudostem circumference of 5 to 6 centimeter of 60 days of total hardening, and it needs to have 4 or 5 active leaves, and interfoliar space must not be less than 5 centimeter, and approximately 25 to 30 height more than 50 centimeter active roots. And the end of the secondary hardening cycle, it needs to have more number of roots at least. So the normal, so this is a picture for the normal bunch that is produced by normal suckers, and this is from the tissue culture banana bunch. So there can be a very large difference produced the bunch from the normal suckers and the tissue culture produced plantlings. And also, apart from rhizomes, you can also use this male flowers. Male flowers can also be used to produce, so that in rhizome you get only one, but in male flowers we can get many number of shoots because there are many number of flowers. So from each flower we can get each plantlet, so that it is possible. This is not normally followed, but this is also possible using male flowers to produce a many number of shoots from a single flower. And also the other like non-defined, undefined media that can be used, like sweet lime juice, tomato extract, sweet corn juice. So anything, all this type of other natural substitutes or undefined media can also be added for the media for producing many number of shootlets. So the problems in banana propagation for the banana tissue culture, the first excessive blackening or browning of the media caused by oxidation of the phenolic compounds. And so this forms a barrier around the tissue and prevent nutrient uptake and also hinder growth. So during the first 4 to 6 weeks, fresh shoot tips transferred to a new medium every 1 to 2 weeks, and alternatively fresh cultures or can also be kept in complete darkness for 1 week. And also antioxidants like ascorbic acid, citric acid at 10 to 150 milligram per liter can also be added to the growth medium to reduce blackening. Or also this antioxidant solution of cysteine can also be used to reduce the browning of the media or blackening of those rhizomes. So transferring to the next to the growth room, are incubated at optimal temperature of 28 degrees centigrade with 12 to 16 hours light, and the temperature needs to be maintained using air conditioners. So the multiplication rate using tissue culture. So starting with the initiation of the first, from one plant with the first subculture at around 50 days, you get around 3 to 4 plants, and subsequent subcultures. It can reach up to say like 11 to 12,000 plants from just one plant within less than 1 year. Right, within 1 year, with less than 1 year, you can get from one plant to around 12,000 plants. Right, so then it goes in for rooting and also primary hardening and secondary hardening. There might be some depth, so at least you will get around 10,000 plants from a single plant, from a single mother plant. through tissue culture. So the varieties, so the different, mostly grand name you see in all the states. So grand name is the main variety that is being propagated in almost all the states in India. In Tamil Nadu, you have different, say like Matti, Sivare, Naipuvan, Rastali, Virpakshi, Nendran. All these varieties are being propagated in India, mostly the grand name bananas. And this gives the income, the normally from tissue culture and sucker propagated bananas, the number of bunches. So around 200 bunches less than the tissue culture banana from the same number of plantlets what we plant in the field. So the net income is it can be more than 30% when we go in for tissue culture propagated plants for the same area, whatever we are going to plant. The production capacity, so the largest producer will be this Maharashtra, and you have this Jain irrigation with a production capacity of 60 million plants per year. So to start a tissue culture industry, so how many plants do you need to produce? First classification. So to start and sell plants, certified plants as a company, how many plants do you need to produce per year? First class you need to produce at least 1 million plants per year, right? Or 10 lakh plants per year to be certified as a tissue culture industry. So in Maharashtra, Jain irrigation is the largest producer of banana plantlets and also tissue culture plantlets. Banana and we have many number of so this data is around in 2010. So now it might have increased a lot, many number of industries. So in Tamil Nadu, we have only the registered Speak Grow and also the Grow More Biotech. Right, so only these companies produce the certified tissue culture plantlets. So for export, for any tissue culture export, so there is a specific packing structures that needs to be followed. So everything needs to be packed neatly. So this does not need to go through the quarantine. Quarantine, the checking so this can be packed in corrugated boxes and then start to export. So the next is the ornamental. We'll be looking at the rose. Propagation of rose is the same. We also follow the same 5 steps that needs to produce, which is used in home garden, cut flowers, and potted plants. So normally, like you see, cutting, layering, grafting, and we can also get some seeds from rose for new cultivars by crossings. And vegetative propagation, which is a predominant technique that is being followed, but this does not ensure healthy and disease-free plants. And it also depends on season and the slow multiplication rates. So we can go in for in vitro propagation. So there is a need to introduce efficient methods for propagation. So the major factors is the choice of explant. What we select for rose, it will be the nodal segment, axillary bud shoot tips. So mostly buds taken from soft wood. It is called as this pencil thickness wood. Right? So the stem, not very thin stems. So it should be of pencil thickness. It should have around pencil thickness from the mid stem region, and the explant length and diameter. Length also like 1 inch, maximum 1 inch. Right? And growing place and the growing shoots in horizontal position and prevent browning of the media and inhibitors of ethylene biosynthesis. Say like the silver nitrate can also be induced so that it can elongate. An addition of growth regulators, the media what we use, the in vitro rooting and age. So all this affect how many plants we get from rose. So for using shoot apical meristems of rose, use MS media with 1.5 mg BAP, where you can get a 94% shoot formation within 6.2 days. And you can get 20 shoots per culture or cutting on MS 1 mg BAP in kinetic, or also using nodal segments, nodal segments, axillary shoots on agar media with 1.5 mg BAP, and in vitro routing with half MS and 1 mg IBE. So using this media can propagate the rows. Again, this is again a different procedure in vitro raised micro shoots. Again, get many number of shoots from the growing shoots. Right? So here you can also cut whatever the shoots you get, and also again replant it, reculture it. So this, whatever the shoot you get, that can be replanted, and you can get many number of shoots. So like induction of shoot bud, or get many number of shoots from this. So from a single explant, you can get many shoots, and from those shoots, again, next culturing, you can get many number of shoots from those. So it can also be grown in bioreactors. So bioreactors in liquid culture. So this rose explants can be put in the liquid culture, so that you can get many number of shoots, which can also be propagated. It's not in your carnation. You can get to propagate other flower crops like carnation, tulips, uh, carnation, lily, then orchids. All of this can be propagated through tissue culture, anthurium, gerbera. So all this are being produced using tissue culture. The next is the 11. Okay. This embryo. First, we will finish the anther culture. Next, so we are looking at. So for in a tissue culture, we need to use specific explant for specific purposes. Right? So in a purpose you tissue culture பண்றோமோ அதுக்கு specific we need to select those explants, right? So for to eliminate virus, so what explant can we use? Meristems. So for micropropagation, nodal explants. So to get haploid plants, right? Haploid plants. So in breeding, so the important time consuming area is to getting the homozygous plants. So to get homozygous plants, we need to backcross it for at least 12 times, which may take up to 8 to 10 years, right? So which is the time consuming process. So instead, within 1 year, if we can get the haploid plants and double it, right, which will be called as dihaploid plants, right? So this will reduce the breeding cycle. So to understand that, if we look at the life cycle of any plant, there is alternation of generations. So in 10th and 12th standard, might have already gone this, there will be alternation of cycles. There will be diploid phase and a haploid phase in plants, right? Sporophytic phase and gametophytic phase, right? So there's alternation of generation in plants where you have the sporophyte, which is succeeded by the gametophytic phase, where it produces the gametes or the ovule or the pollens. So this is known as the alternation of generation, and the sporophyte phase. The spores or the pollen is produced by meiosis within a sporangium. With the sporangium, which will be those anthers. Right. So the chromosomes pairs are separated to form single spate, and spores are haploid, develop into haploid gametophyte or those pollen grains. And this gametophytic phase, the gametes produced by mitosis within the antheridium, which produces sperm, and in the archegonium, it produces the eggs. And this fusion of this male and female gametes. So these are all haploid, right? So the sperm, the pollen will be haploid, and the egg will also be haploid. And the fusion of these two will give the diploid plant, right? So this, the pollen alone cannot develop into your plant. It can develop into your plant. So we can, by altering the hormones, we can make this either this or this alone alone to produce a plant, go into this sporophytic phase. So this is a gametophyte. We can make this gametophyte to reach this sporophyte phase, right? But this will not produce like viable seeds because there is only one end set of chromosomes. So for producing viable seeds, you need to have two, two one end set of chromosomes that needs to fuse that will produce viable seeds, right? So if we produce a plant from just one end set of chromosomes, it will not produce viable seeds. We can get plants, but it will not produce viable seeds. So அதுக்கு என்ன பண்ணலாம்? So in the life cycle of a plant, you get the flowering plant. So you get the stamen or the antheridium. So we get the microsporocyte. So inside this micropole, which is called as the pollen mother cell. The pollen mother cell. So the anther develops, which are microspore tetrad phase and the triad phase. All this phases. So all this. So each of this blue arrows or the diploid stages, and the red will be those haploid stages. So this diploid stage will, by meiosis, will be converted into this haploid stages. So from this meiosis stage, from right here, it started producing this pollen grain, and we cannot directly use this pollen grain because this has a very thick wall, pollen wall. The pollen wall will not germinate in the medium. So we need to select those pollen grains before this, after this meiosis, but before it generates into a pollen grain. Upper where this exine or intines. So if you remember this pollen, you have this exine intines, very hard pollen wall, so which cannot germinate onto the medium. So before that pollen wall forms, we need to select such a stage which can germinate or produce cells in the normal medium. So you get the pollen grains, and when it germinates onto this stigma, so this produces the pollen tube, which fertilizes the egg with the embryo sac and produces fertilizes it and produces the embryo, which is in the seed, and produces the young sporophyte. Or it can also go through this egg phase, the ovary or the gynoecium. The same phase follows. You have the megasporocyte, where you get the egg, and which are also all are haploid. So either we can take this part or this part, right? So the first is the anthers. So mostly will be because anthers we can have many number of anthers which can be used for production, but eggs we can get only one per flower, right? So that is limited. So there are in the development of this gametophyte. So which stage we can select for development of those callus or plants? So each anther which has this microsporangia, and this microsporangia contains this microsporocytes or the pollen mother cells. Microspore pollen mother cells that undergoes meiosis, producing 4 tetroid or haploid microspores. And each of this 4 components of the tetrad develop into this pollen grains. Right. So we need to select before the pollen grains are formed. So if we look at this anther, so you have all this the tapetal layers, and you have this microspores in between this the vascular bundle and also this connective tissue. So we need to select before this pollen mother cells becomes the pollen grains. So this in the pollen mother cells, when it develops, it can be in the tetrad stage or the triad stage, three cell stage. So it undergoes meiosis 1, meiosis 2, which gives rise to this 4 haploid cell tetrad, which releases the 4 microspore, and this enlarges into microspore by evacuation or uninucleation, and with the highly cytoplasmic nucleus with the central nucleus. And you can also get a dinucleated microspore with the first pollen mitosis and vegetative stages and vegetative cell, and also the generative cell. So when it divides, it can divide into this vegetative cell and also this generative cell, right? So you need to remember this. So next, rather this vegetative and this generative cell. So this will differ according to different plants, right? So over plant, over the vegetative cell before the pollen grain develops, right? Vegetative cell and the generative cell. And the tapetum cell layers, the cell layers which separates it from the tapetum wall also degrades, which gives rise to this trinucleate pollen, what we get here. So it is important to select the specific stage for culturing of this plantlets. So this tapetum layer degenerates after the microspores are formed, and these are all fully absent before anthesis or the dehiscence of this anthers. So the androgenesis, how we can develop androgenesis is development of plants from anthers. Anthers is androgenesis, haploid plants from anthers of microspore. First discovered by Guhan Maheshwari, right? Should not forget this. Again, for an examination point of view. And the critical factor is the change in the developmental pattern from the mature pollen grains to embryogenesis. So these are all haploid cells, which is a gametophytic phase, and which can develop only after fertilization. Only if it encounters the egg, it will again develop into a plant. But we need to convert this mature pollen into this embryo, into an actively growing embryo. So how we can do that? So that is androgenesis. So production of haploid production using androgenesis. Haploid is having the same number of set of chromosomes as the germ cell, or half the diploid number of somatic cell, having a single set of chromosomes. So this process inhibits the typical gametophytic differentiation, and instead it allows the cell division and regeneration and production of plantlets to occur, and double haploids can also be produced from in vitro by producing this in vitro by doubling the chromosomes. So these are all not called as diploids, right? So normal plants are called diploid plants. So these are all called as dihaploid plants. So, which are all homozygous plants. But normally in the field, the conventionally, this can also be produced by delayed pollination or irradiation of the pollen, killing of those pollen grains, or distant hybridization. You can also produce haploid plants. But these conventional methods are cumbersome, laborious, time consuming, and also not efficient. So this is done in tissue culture by using an excised anther and microspore culture, or also using ovaries and ovules. So in anther culture, using this androgenic haploid is the most promising method that is being followed for many of those plantlets. I use this microspore or uninucleate pollen in the early developmental stage. All right, so we need to select that specific developmental stage so that we can convert the single N, 1N number of chromosomes to divide into get many number of cells. Can be induced in vitro to form a plantlet. So for the most plant species, early to mid nucleate stage of microspore is most responsive, and it can also be induced in tetrad stage and also the binucleated pollen stage, where so each of the flowers we need to go in for cytology studies. So what is the stage of those pollen grains? The size of the flower bud. Looking at the size of the flower bud, we can decide what would be the pollen stage inside the flower buds. And also pre-treatment or with the cold treatment. So this can also induce the androgenesis, like cold storage at low temperatures. So this increases the number of microspores which arrest the first mitosis, and also the starch production is blocked, so that the pollen formation is blocked, and also retards the aging of the anther wall, and also prevents degradation of the tapetum matrix, formation of all those exine enzymes in the pollen grains, which also allows a higher propagation of microspores, and the developmental pattern can change from gametophytic to the photosphytic space. The cold treatment can be from 3 to 6 degrees centigrade for 3 to 15 days. So starting from say like for 14 days at 8 to 10 degrees, you can get around 7% response of those anthers. So by giving this cold treatment, weak or non-viable anthers, microspores get killed, and only those viable, strong pollen grains or anthers can be used, which will give rise to the plants. So this also retards aging of the anther wall and use of liquid medium. The media can be a liquid media in a 3 So the two modes how we can get plantlets. Plantlets from anthers is from direct embryogenesis from the anthers. This can directly produce the embryoids and also the haploid plantlets, or it can go into indirect where it can produce the microspores can produce callus, and from that callus this haploid plantlets. Plantlets with or without roots can also be produced. And all this are haploid plants, right? Because these cells are haploid. So this is again the pictorial representation where you have this heterozygous diploid plant, heterozygous diploid plant, take the flower bud. The anther can be produced, can be kept in the media where this can produce the direct embryogenesis, producing haploid plants, and these plants producing haploid plants, and or indirect organogenesis producing callus, and this can give rise to the haploid plant. And this haploid plant cannot set seeds, so this needs to be treated with colchicine, which can become a diploid plant, right? And which where the seeds set will be fertile. So this is the important part, the pathways to androgenesis. It can be in 5-mark questions. So what are the pathways that this anther can take up to give this pollen grains, and which stage we need to select? Because it is different in different plantlets, say like Datura, Nicotiana. So there are 4 different pathways through which it can take, giving rise to the development of this multicellular plantlets. So in pathway 1, the uninucleate pollen grains. So this can divide symmetrically, which yield 2 equal data cells, both of which undergo further divisions, like in Datura. So like if you look at this, so these are all thus 4 different pathways. So you need to remember this pathway, mostly in 5-mark questions. Right, so you have 4 different pathways, 1, 2, 3, and 4 different pathways of where you get this multicellular pollen grains, which can give rise to the plantlets, right? So the first one is you have 2 data cells, which can both divide equally, right, to give rise to equal number of cells, and then you get multicellular cells, right? So this is pathway 1. Or pathway 2, in some other cases in Nicotiana, the uninucleate pollen divides unequally, on the generative cell. So you have 2 types of generative cell, and the generative cell immediately degenerates or after undergoing 1 or 2 division, and the callus or embryo originate due to the successive division of the vegetative cells. So this generative cell degenerates, and only the vegetative cell give rise to the multicellular plantlets, right? So in Pathina, so you have this vegetative cell and the generative cell. Generative cell degrades, and only the generative cell, right, multiplies to give the generative cell multiplies to give this multicellular pollen grains and give rise to the multicellular plantlets, right? So the pathway 3, the plantlets in Hycocymus, mustard plants, right? So the pollen embryos originate from the generative cell alone, and the vegetative cell either does not divide or divide only to a limited extent, forming a stick-like suspensor-like structure. So only the generative cell alone divides. So when you look at this, so only the generative cell divides. So this again like degenerates into a suspensor, like or stick-like structure, and only the generative cell divides to give rise to the multicellular cells. And the pathway 4 in again in Datura, the uninucleate pollen grains divide unequally, producing regenerative, generative, and vegetative cells. And but both these cells divide repeatedly, giving rise to unequal number of cells, unequal sizes of cells, which contribute to the developing embryo or callus. So both these cells, generative cells and the vegetative cell, both divide unequally, to giving rise to many number of unequal sizes of cells, which give rise to multicellular pollen grains, and this can give rise to the direct embryogenesis or through the callus phase, indirect embryogenesis. So these are all the four pathways by which this pollen grain, starting from the microspore, can give rise to an entire plant, sporogenesis. So diploid station. So this cannot give the plantlets cannot give rise to fertile seeds, right? So this will not produce viable seeds. So instead, haploid plants from either anther or ovule culture. So this can grow normally under in vitro condition and go up to the flowering stage. But viable gametes are not formed because there will be no separation of chromosomes. So there will be no meiosis, there will be no separation of chromosomes, so there will be no viable gametes are produced. So there is no viable seed set. So in order to obtain fertile plants, the haploids must be double, which are called as double haploids. Double haploids or dihaploids, not diploids, right? So these are all called as dihaploids. So this is the most sought after technique in seed industries now, right? So seed industries needs to produce seeds in a quick time. So instead, if we go in for the double haploid or the dihaploids, we can get immediately the homozygous plants within a shorter time. So chromosome doubling can be done by treatment with colchicine by immersing those very young haploid plants in colchicine, 0.4% colchicine for 2 to 4 days, or spontaneously, like in maize. It can occur in haploids at varying levels, right? But it's not very efficient. Many systems are using visual observation to detect spontaneous dihaploids. So usually going for this colchicine treatment. And the culturing of this microspores. So this collect the anthers at both leaf stage. So before the flower opens, so collect those flowers, incubate at 4 degrees centigrade for 4 to 10 days, sterilize with mercury chloride 0.1% and dust in vortex and collect the microspores which is forced out of the anther wall, filter it and centrifuge it so that it settles down and add this pellet onto this semi-solid medium. So the viability of those microspores can also be checked with potassium iodide. So potassium iodide will stain starch grains, right? Iodine will stain starch. So this more of starch grains will give more viability, right? And so if there is no staining, it means the pollen grains are dead. Or also uninucleate stage staining with acetocarmine. So this will stain the nucleus so that we can see the amount of nucleus present inside. And also can culture in liquid media instead of semi-solid media. The uses of haploid plants, homozygosity can be achieved in the quickest possible way. Then the generation of genetic segregation is simplified, and recessive genes are not being masked. So obtaining recessive genes, homozygous recessive genes will take many number of say like 12 to 15 cycles of pollination to get recessive homozygous plants. So this can be gone by this androgenesis we can get within 1 year. Complete homozygosity in step is compared in one step compared to 7 to 8 generations of self-ping by normal breeding procedures. The selection is much more easier and efficient. Say like for example here, say like in F1, if we cross like the heterozygous homozygous line. So in F1, you will have the combination of all this, right? So the gametes, you can have the all other gametes and the double haploids. In the double haploids, this will be exactly doubled, right? The RGT will be RRGGTT, right? So here, within one step, you can get the recessive homozygous plant within one step. But in normal breeding process, it will take 7 to 12 years to get homozygous plant. Reduction and selection of recessive alleles and easy selection of characters. So also facilitating mutational breeding. Uses mapping quantitative trait loci. So because of easy production of double haploid lines, like QTL mapping, which also will be much easier, and the cultivar development is also much earlier, right? So normally comparison of a conventional breeding program with utilizing haploidy, the normal conventional breeding. So after we produce F1 in the first year, it can take up to 6 to 7 years until you go fund for evaluation. Right, you need to go in for back crossing every time for 6 to 7 years before you go in for evaluating the plants. Right? But in haploidy, within 1 year, you reduce all that 6 to 7 years into 1 year. Right, and you get a dihaploid plant, and you can under all of the process evaluation, field evaluation, and licensing of that and releasing will also be the same. But this breeding process is entirely reduced. 8 to 10 years will be reduced when we go in for the haploid. Right. So this will be this use of using androgenic haploids. So the next. Okay, these are all those. So what are all those important factors? Genotype, stage, physiological stages, culture media, incubation condition, and the wall factors. All this influence the success. The problem with haploidy is low yield. Like you saw in the table, you can get up less than 10% of yield from the haploid plants. Only less than 10% of those pollen grains germinate to give rise to the scalus or embryoids. And 70 to 80% of embryos that are produced are incapable of germination due to structural, physiological, biochemical abnormalities and the instability of the genetic material. Because this changes, because you have only one set of chromosomes, as even a single mutation can give rise to mutation in the plants. So instability of the genetic material. So this causes mostly the albinism, white plants without chlorophyll. In those plantlets, especially in haploids, you will get more than 50% of those albino plants. Very response in some, very low response in some species of cultivars. And Colchicine doubling is also not successful in many of those species.